There. Okay, so what we're talking about today is we're talking about mutations. We're still doing a crash course. I'm thinking that next week we'll actually begin chapter 2. So for those of you who are waiting for when we begin the book, that will be coming up very, very soon, and that's where where the actual class material begins. This stuff that we're talking about now this is still part of the class. It's still very important, but the reason why we're spending so much time talking about this and about some of the other things, and about the biology and the central dogma of biology. I talk about all these things because I want you to be aware of some of these concepts. These are the pieces that kind of like the pieces of how the discipline actually works. So like the fundamentals of the discipline, and so that's the reason why. So I'm looking at the page fit here. Anyway, hopefully this will. Let me see if I can get this thing to fit in here perfectly. Maybe not very well. Anyway, I have to make do with that. Anyway, so these are the slides, and so I'm going to spend some time now talking about something which actually I think is quite interesting. We're going to talk about mutations, and mutations are actually, you'll be surprised, mutations happen more often than we think. And this was actually a surprise to many of the biologists out there who were working on mutations like back in the 50s, 60s, and 70s, where they just realized, or they just thought that mutations didn't happen until it was like, unless it was like some strange radioactivity exposure or radioactive exposure or something, but they didn't happen to DNA unless the DNA was actually forced into being mutated. But we know now that that's really not true, and mutations happen seemingly all the time. And so we begin this conversation with the whole idea of, or the whole question of like, well, what is a mutation? What am I talking about? I'm using this word. Well, a mutation is really a misspelling, if you will, a misspelling in DNA. Now I say a misspelling, but really what I mean by that is that the DNA looked like one thing, it did it, and then it copied itself a couple million times, and then when next I saw it, it was different. Something had changed. So a mutation really doesn't necessarily mean a bad thing, but it just means that it's a difference, it's a change. It's not what you're expecting to find. And so you'll find that mutations actually happen fairly regularly. I mean, there's a lot of reason for that, and we'll talk about that here, which is actually very interesting. But DNA seems to change itself all the time. And I will say this, though, that mutations are actually fairly necessary when you're talking about, when you're talking about evolution, because the DNA cannot or should not just continually be the same thing from generation to generation to generation to generation. And the reason for that is because if the DNA is not allowed to change by some means, in this case a change is a mutation, if it's not allowed to change, then that means that the organism will not be able to adapt to new pressures in the environment. And so the mutations are really adaptive processes which help the organisms to embrace changes and to make changes. The whole reason that we have better and better eyes, for instance, in our heads than what we had when we were first, fish-like creatures, is a result of mutations that happened under the influence of environmental pressures. Every time we had a mutation, if that mutation helped us to see better or helped us to do something better, then it stuck around because it gave us some competitive advantage. However, if that mutation did nothing for us and made us sicker or weaker or less able to compete, then we would have died, and with our death, that mutation would have gone too. And so you can see that mutations, if they help, then they stick around. But that's not always the case, though. If they don't, basically the rule is if they don't kill the host, then they stick around. Anyway, I wanted to say, though, that a common process here that good morning. That during replication of the human genome, a typo may occur fairly regularly. And by regularly, I'm saying basically every 100,000 or so nucleotides. Now, 100,000 seems like a big number, but actually in the space of DNA, in the DNA space, 100,000 is really not that big at all. That's like the number of cells that you might kill just by snapping your fingers. And so it's you can see that's a fairly regular thing that's going to happen. So here's another interesting statistic here. It says about 120,000 typos every time one of our cells divides. So you're basically thinking about these mutations happening fairly regularly, and that's just a fact of life. But the thing is, though, that there is help. We have most of these mutations are repaired. We have actual systems that have evolved to determine what it is that's gone wrong here, and then something that fixes that incorrectly spelled nucleotide here. So one thing I should also say here is that these mutations are neutral. There are no consequences. So if you're a computer scientist and you're interested in hearing this example, then maybe this is the best one. If you were to take a piece of computer code and you just put this on you open it up on your screen, you open it up on your editor, and you just began like pushing buttons wherever the cursor was, and then you tried to run it. It's not going to run. Chances are, it's not going to run. Because why? Because we've introduced some syntax change to the code. However, if you are just pushing buttons in an area of code which is commented out, which means there's no syntax, the compiler never goes there. The Python doesn't read that area of code. It's a comment. Then you can push as many keys as you like, and nothing is going to change. It still runs. Everything is fine. That's exactly what happens for instance, when you have a neutral mutation. Your mutations are happening in an area of code which doesn't really do anything. When I say it doesn't do anything, I mean it doesn't code protein. So it's not like the active code in a piece of software. So if you have a mutation there, it's probably going to be neutral, where there's no consequence, and you'll never know that there's a mutation that ever happened. And otherwise, if a mutation happens in a place where the code is being written in a piece of software, then it could be, I mean, there's a non-zero chance, but it could be that by pounding all these keys, you might introduce some change to your software which fixes it. I mean, it's arguable, but it could happen that just by pushing the buttons, you might somehow do something that's going to fix your code. In fact, in mathematics, this is your, a million monkeys at a keyboard, and they would say that a million monkeys at a keyboard or at typewriters over the course of an infinite amount of time, you're surely bound to have at least one monkey who's going to be typing up the complete works of Shakespeare, in an infinite amount of time. So what I'm saying is that it could happen that you could get something beneficial from a mutation in this case. Your mutation happens to a piece of protein, or a place where protein is coded, and the mutation makes some benefit. Although some other mutations are harmful, where that means that just like if you're changing your code in your computer, on your computer software, you might actually introduce a bug by just randomly pushing buttons. So you get the idea, though, that their mutations are really random, and they just happen seemingly anywhere. And sometimes they help. I would say sometimes they do help, but sometimes I'd say most of the time they probably don't. But that is the thing, though, the vehicle that allows the DNA to evolve. That's where the changes can come from, one of the ways. Anyway, I wanted to give you some ideas about what kinds of mutations are out there. This is a chocolate chip cookie recipe, and by the way, you can cook this up. You can bake these cookies. I've done that with my daughter, and she said that they were actually fantastic. But so it's a good thing. It's a good thing. It's a win-win. You can study bioinformatics and also make some cookies. So anyway, here is a wild type recipe. These are the instructions for how to make cookies. How many ingredients do you have to have? What amount of those ingredients? How do you mix them together? It's all that stuff here. But when we introduce what's called a mutation, when you look at this recipe here, you don't necessarily see that there is a mutation. It's there, but you don't see it. Where is that mutation? Well, you have to really, in bioinformatics, how you'd find mutations if this were code, DNA code, is you'd have to look at the original, and then you'd have to compare with the thing that you suspect has some kind of mutation. Here, I'll just tell you where it is. It's in this 1 cup 2 STQCKS butter, which is right here. So this is what they call a point mutation, where for some strange reason, and maybe it's not completely understood why that happens in many cases, but one of the pieces or one of the characters in this recipe, or a base in DNA, just flips. It's something different. And so in this case, you can see that if you're reading this recipe and you see a STQ, it should be like sticks of butter. The I has gone to a Q. You can see what it was before. Sticks of butter. But you can kind of get an idea, because you're familiar with the word, you have an idea of what that word would have been. And so that's kind of error correcting in a way. Your genetic material can be read and understood by some of the biological machinery in your cells. and it can look at some of these words and it can say, oh, you know what, that's a typo. That Q should have been an I, so therefore I'll go back and switch that. It just recognizes these things. That's kind of how the recognition system works. We do the same thing because we know this language. We know what that word should have been. So that's a point mutation. Now here we are back again at our recipe, and there's another mutation here, and that mutation is going to be, I'll tell you right down here, it is a point mutation of not, it's no longer, what was it before? It's no longer 9 teaspoons, or sorry, it's no longer, sorry, it's 1 teaspoon, here we are, 1 teaspoon of baking soda. This mutation is now 9 teaspoons of baking soda. So you can see that that's a more serious kind of mutation, because unless you were familiar with this recipe, unless you knew something about baking cookies, that you knew that there's no way you'd ever use 9 teaspoons of baking soda for such a thing. If you knew that it was supposed to be 1, then you wouldn't catch that error. And you might just go ahead and make those cookies and start adding 9 teaspoons of baking soda, and your cookies would be ruined. So that's a different kind of mutation where you can't really pick it out. You can't really see it coming. Now here's another one here. I apologize for all these things here. It works better when you can click through these things with a laser pointer, but I just left them the way they are in this slides. Excuse me. Okay, here's the next one here. The mutation is present. Where would that mutation be? Well, it's going to be, you can see that, look at the bottom of this thing here. It says preheat at the very, very bottom of the recipe. That doesn't make any sense. Unless I knew the mechanics now of making a recipe, unless I knew how to actually, I mean, I have the ingredients in hand, but unless I knew how to actually use those ingredients, then I wouldn't catch this error either. And so here's this is a what they call an inversion type of mutation, where the code is still correct. It's just in the wrong place. So the wrong order. So the preheat to over 375 degrees Fahrenheit, that should not be at the end. In fact, originally that should have been right under the ingredient section in the instructions. And then, once that step had been completed, then you could start loading the oven with the cookie mix. And so what I'm saying though is that you have these mutations which, while they seem very minor, they actually create some very big effects. And so you can see that the cookies now will never cook, because you're putting the cookies into the oven, and maybe there's some kind of a catch that says wait for the cookies to bake before taking them out. So therefore the cookies are in the oven indefinitely now, and we can't get them out. And then maybe we can't preheat unless there's something else going on. So the baking can't happen until the oven's been preheated. Okay, so let's go back to our recipe again. And I think this time we have the preheating where it should be. There's a mutation now present in this recipe, and this is a substitution type of mutation. And what it was before was we were supposed to have chocolate chips, 2 cups of chocolate chips, but the mutation now has become 2 cups of butterscotch chips. I'm not sure if that sounds good or not. It could be. I mean, I'm open to things. I mean, I'm open to things. But what I'm saying, though, is that this is no longer a chocolate chip cookie mix, because the thing that makes some chocolate chip cookies is not in there anymore. So this is now a butterscotch chips cookies. Now, maybe, for instance, if this were DNA, maybe chocolate chip cookies, the DNA for chocolate chip cookies was no longer effective. In the environment, the restaurant environments where there are pressures to force this recipe to evolve and adapt, maybe those pressures were just too much, and the word chocolate kind of got squeezed out of the recipe here, and so it became butterscotch. And then maybe the clientele in this environmental restaurant appreciate butterscotch better. So now butterscotch has more of a pull, and therefore it has a better competitive advantage than chocolate chip. So maybe by this substitution, which was completely haphazard and completely random, maybe switching chocolate chip to butterscotch was actually a good thing. Why not? I mean, I like butterscotch every now and then. But what I'm saying, though, is that mutations, they don't necessarily have to be bad. In the case of where the preheat and the order of things, the inversion, that could be bad. But the other ones are not necessarily bad mutations. So here we have different kinds of mutations to think about. We have point mutations, which are mutations that happen at specific areas in the code. Then we have these inversion mutations, where chunks of code are kind of moved around. maybe maybe a chromosome or something. Yeah, a chromosome is like maybe reversed in some way. Pieces of code are reversed in some way. Something has, I mean, it's like the code is correct, but it's just that it's been switched around, or it's been like kind of taken from here, reversed, and then it's pasted someplace else. Then we have a substitution mutation here. It's like when one base is substituted for another. It's like a substitute teacher here, where the teacher can't come into class, so she has somebody coming in to take her place for her just for the day. But in this case, the base will come in and be replaced, and it will not ever be unfixed. Now, these are substitution mutations can generally be caught because of the biological machinery that actually does the coding, or that works with the DNA, is able, in many cases, to recognize that this is a typo. There's something wrong here. There's a typo that I can fix this. But if it's an inversion mutation, it's not always as easy, because the bases are exactly where they're supposed to be. They're just not in the right place. It's like they're not. It's like they've been switched around or something. And then point mutations, again, those are sometimes hard to figure out, because unless you know what that word is supposed to be, unless you recognize what that gene was supposed to be before, sometimes you can't always figure out what that is. And so what I'm saying is these mutations do happen. Here we are with types of mutations, where we have insertions, we have deletions, and frame shifts. We're going to talk about those coming up here. But insertions are where you have a sequence of DNA and something else is like thrown in there. So if you went back to our recipe where we'll say, here's our chocolate chip cookies, an insertion mutation would be grab, I don't know, 2 buckets of honey and dump it into the recipe. It's like it has nothing to do with the recipe. It never was there. And here we are now having this line of of our recipe. Dump all this new stuff into our recipe. And you're thinking, well, maybe that works. I mean, I don't know. Maybe, is that a mutation? Is it not a mutation? And the thing is that sometimes you can't tell. So in insertion, sometimes you just can't tell. Deletions, where, for instance, you'll go back to the recipe here and you'll just take something out. So I'm looking at 2 large eggs. I'm just going to take that line out. I don't have any eggs today, so let's take it out. But the thing is that if I were to take out the eggs, I mean, eggs go into recipes for a reason. It helps everything stick together. It helps the flour to work, to stick everything together. And so if I remove those eggs, then the cookies don't look like what they're supposed to look like. If I take out a chunk of DNA from a piece of code here, then maybe the protein's not going to look like what it's supposed to look like in order to maintain health. And so you can see that there are some very serious problems that can happen. And when something has been deleted, by the way, it's very hard, both in biology and in regular life, to look at that something and say, oh, something's missing. Something's missing here. There's a piece of this code which is missing. There's a piece of, suppose I gave you a piece of source code today, right now, and I removed a comment which would have helped you to run this thing. You may not know how to use this code, and so because I've removed it, you don't know that it was there. You don't know that it ever was there, and you don't have that knowledge, and so sometimes you can't. You can't. There's no way to figure out how to move forward. Another type of mutation is called the frame shift mutation. This is actually very interesting, where, we'll talk about that in a second, but actually it concerns when you're creating proteins. And the idea is that when you're creating proteins, DNA bases are read 3 by 3 by 3 in codons or triplets. And if one of those triplets is removed, then your reading frame has been kind of offset by 1. So now you're reading 3 bases at a time, but they're not in the same order. So the one that should have been the first base, and the one that should have been the second base, and one that should have been the third base in your reading is no longer in that order. So you have, for instance, now you're taking away the first one, the second one, and maybe the third one here. And so now you're reading these 3. If you can see my fingers here. I'm not giving you guys the finger. It's actually, I'm using my finger to show something. I'm looking at myself in this picture here. It's the same one you see up there. And I'm thinking, this just looks really weird. I'm showing my fingers. Sorry about that, guys. Anyway, I have a slide that talks about the frame shift mutation. It's actually quite interesting. Anyway, so let's talk about these mutations then. In terms of DNA, how does this mutation look? So here we are with a point mutation where what we see is this T, you see T, A, C, T. The T has miraculously become a C. And I mean, it could have happened for a number of reasons. Radiation, for instance, will do that to you. You can get all kinds of, these charged particles could actually fly through space, I guess, or neutrinos and those kinds of things, or other kinds of I guess electrical particles or whatever. They're particles, energetic particles, from I guess X-rays and those kinds of things, microwaves. They could fly through and potentially knock out a base, which is then replaced by your copying mechanisms, and the base is not the same as when it's copied over. This is like what a point mutation would look like in protein. You can see that when you're reading, what I was saying before, is like when you're creating or translating DNA or RNA rather to protein, you're going 3 by 3. So you have each base is read. Actually, 3 bases are read at a time. Those bases are then translated into specific codes that create the protein. And you can see that these are all CAT, CAT, CAT. That's the original code. But at the place where one of the CA or the CATs, where the A has become a C, you can see now that the protein has changed. It's not the same protein any longer. And so that in itself causes these big problems. A lot of people, when mutations were first found, they thought, well, what kind of problems could we possibly have from mutation? So what? The DNA is different. But then when you remember that DNA actually gives us the protein, the stuff that we're made of, then the problems could become much more grave. And so this is exactly the situation where now your protein is, at that particular amino acid, it's different from what it normally would have been. Interestingly, we'll have a conversation later on about sickle cell anemia, and it's caused by similar types of very subtle mutations, but the consequences are very, very impactful, and they cause a lot of suffering. Anyway, so here we have with a substitution here. This is another kind of a mutation where we have a substitution where a substitution, I'll just read this, is a mutation where one or more bases in the sequence is replaced by the same number of bases. It's like you're taking out a word and replacing it with a different word. So I guess in the case of these sequences here, you're basically taking out this, G or TGC, and you're replacing it with GAT. It's like it's the same size word, just pulled out. Now, this substitution is not going to change the frame shift. You're still going to be able to read 3 at a time, 3 at a time, 3 at a time, 3 codons at a time here to create your protein, and you're not going to be out of register when you're reading through to do your translation. However, the protein that you would, or the amino acids in the protein that you're creating here, which would have been, I guess, I'll just say TGC, that protein is now going to be different. So that amino acid is going to be different. And so if that protein is sustaining life, and somehow that addition or that change makes the protein less able to maintain survival or maintain life, then that's a bad thing. Now, this is another type of mutation. This is what they call an inverse or an inversion type of mutation. And what happens here is where, for some reason, the DNA is cut by some, there are restriction enzymes that do some cutting here that may somehow get they may be deployed in the wrong place, or maybe there's a virus that comes in that actually cuts up the DNA. That happens. There are certain viruses that break into DNA and they attack just by cutting. And maybe before they were neutralized in your cell, they were able to make 2 cuts, and somehow that piece of code came out. It was floating around for a while. Something came along and tried to repair the DNA by saying, oh, wait, here's that piece that belongs right here. But unfortunately, they put it in the wrong way. And so that's what they call an inversion. And so you have the code is in the right order if you think. It's like if you go if you're going the other way here, it's in the right order. But it's just when it's in the whole sequence here, it's in the wrong order. So by itself, that little island is correct grammar, but it's not correctly placed in the full sequence. So that's an inversion. Another one is the insertion, and these are actually more dangerous, the insertions here, because when you have an insertion, this does actually interfere with the codon reading. That means that you actually do get these frame shift mutations where you're creating proteins from this code here. When you have one extra base in there, that means downstream from that one extra base, as you read in 3s, you're going to be reading the wrong codons to create that protein. So here, for instance, we have a T, which was then changed, or that was changed with or a C was then kind of superimposed between the C and the T where it would have been before. You just basically shove something in, but you can see that because you shove something in, then that means that your reading to create your amino acids now is out of register. So this is a frame shift mutation. And we'll talk about that in just a moment, I think it's at the end of these slides. But I think those are the most frightening mutations. This is a deletion, where now I'm just going to the DNA and I'm just going to just plot a base. And again, it's going to change the protein amino acid that's created at that particular place. It will change, it will create a frame shift mutation, because again, the reading of these 3 codons by 3 codons is going to be out of order because something is missing, something's missing. But here you're just taking out the T and just removing it, and you're just sliding everything over, and so it's as if T was never there. And that's the hard part about these mutations, deletions and insertions, and that is that unless you were familiar with the sequence before, you don't really know, you can't really pick out that this is an error. Unless you knew that the sequence had no T in it before at that particular location, you probably would miss this. And some of these mutations are missed because it's the biological machinery just doesn't see that. Now, these are the most spooky mutations, I claim, and that is these are these frame shifts that I've been alluding to. And so frame shifts. Let's just go back to this thing here. Remember when you look at the if you look at this sequence here, AUG, ACG, this is a piece of messenger RNA. It just comes from DNA. The U's were T's at one time in DNA. When DNA went to RNA, the T's were thymines. Now and then RNA they became uracils. Anyway, but still we have their U's. And so when you read a piece of DNA here, you start with this M, it's always methionine here, that's always the beginning of these proteins, it seems. But you have M, T, A, G, P, and then where you have this thing called the frame shift mutation, that's where you shove something else in there. Like so this, in fact, on the bottom here, for this DNA, we've inserted a G between those 2 A's, where you can see that bump in the code. Now, what that does is, in itself, it's very minor, but because you put that gene there, it upsets the entire reading register of the mechanism that goes through the DNA or the RNA reading 3 by 3 by 3 to figure out how to place the amino acids. And so by adding just one little base in there, you're going to be upsetting that reading frame, and that's why they call this the frame shift, because now the frame that you're using to actually kind of figure out how to build your protein is going to start giving you just nonsense. The code is correct, but because you're not reading them where the first codon, the second codon, and the third codon, where that order has been maintained, you're going to get nonsense protein. Well, potentially nonsense protein. But you'll get a protein that's not necessarily going to be helpful. Now, the scary thing is that these types of mutations happen all the time. And so, I mean, if you look at your hands should look okay. You've got fingers, you've got eyes, your hair is growing. You're not sick. You guys look healthy. Congratulations, you look healthy. But what I'm saying though is that because you have these kinds of mutations that happen all the time, and it is scary when you read these things. I mean, if you're looking through the book and you're looking at the sections that discuss mutations, or any book, for instance, it is scary. And you think, oh my gosh, all these things, I have no control over that. How can I protect myself from mutations? Well, relax. You do have special systems inside your body that protect you from these types of mutations. But the frame shift mutations, though, if there's no mechanism that will save you and stop you from getting these things, then these could be like really life-changing mutations, just like that. So it's almost an entire change of your protein that comes out of this particular DNA, just because of one base. Just because of one. And in fact, when we read about something called cystic fibrosis, it's like another simple mutation. It's not a frame shift, but it's a simple mutation, like just one base, which changes the course of life almost entirely for somebody. It's like one simple little base just changing has now made the difference between living a regular life and living a life with cystic fibrosis. We'll talk about that. I think that comes up, is that next chapter? Or is that the third chapter? I forget. Anyway, but we will talk about that. So we ask ourselves, like, well, where are these mutations actually coming from? And how can we figure out more about how to stop them from happening? Well, they usually happen during cell division, which is something called meiosis, where a cell is dividing, and you have all these things happening to the DNA, which basically means that the DNA has to be unraveled, it has to be cut, it has to be copied. All these things have to go on, and then once you've copied once you've unraveled the DNA, you have to basically build the daughter strand on each of these sides here using that semi-conservative model of DNA replication that we were talking about earlier. But you're really building the DNA, and when the DNA comes apart here, you only have one strand, and so one strand is the template. So if something happens to the template on this side or the template on this side, that means that you don't have two templates to kind of figure out what the direction is to go. You have one template on each side here, and when you have one template on each side, if something happens to that template, just like if something is cut and you put something back in the wrong way, you can't see that from this template because you only have one copy of this. If you had the DNA helix together, then you could compare both sides and you'd say, oh, this isn't right. This is supposed to be like this. Or you might be able to think, well, I don't know which side is correct, but I know that they don't agree, so there's something wrong. But what I'm saying is that when you have cell division, the DNA comes apart and you have two templates floating around, and those two templates actually do create a possible environment for all kinds of bad things to happen, where things can be mixed. So this is what they call the polymerase here. So your polymerase is really it's like when your DNA is being copied, you're working with polymerases here. A DNA polymerase is an enzyme that catalyzes the synthesis of DNA molecules. So what you're doing here is that you're taking your DNA. The DNA comes apart. You have a strand over here, your parent strand, and you're trying to build your daughter strand. And basically, what that is this thing is floating around in space, and then you have some mechanism that comes through and it just basically places complementary bases on the outside of on the other side of the strand, and that's how you build the daughter strand. So you can see that here, what's happening is that where the red star is, as you are building your daughter strand, you're trying to put an A connected to where a C would go. Well, we know that C and G's are pairs and not A's and C's. And so this A is being incorrectly placed. But the mechanism that places that base doesn't necessarily know that that's a mistake. And so there's another piece of the mechanism here that actually does check to make sure that there's no spelling mistakes. And it says, you know what, I find that this A was not supposed to be connected to the C, so therefore I'm going to stop the production, I'm going to pull off this A base, and I'm going to find a G and put it where it's supposed to be, and then I'm going to continue. And so you do have these kinds of mechanisms, and it's when I see these things, you can't help but smile thinking, this is the thing that's saving us. But it's not perfect, though. It's not perfect, and you do get mistakes that are introduced to your DNA. And sometimes these mistakes are grave, and sometimes they're good. And sometimes the mistakes will help you to have some competitive advantage. Maybe, for instance, you have a mutation in your brain that makes you super intelligent. That's not really what I would call a bad thing. And you always hear about these shows where somebody is like, they're zapped with with some kind of electrical jolt, or they're in a storm and their brain gets zapped, and somehow they have a genetic modification in their brain, and suddenly they're like superhuman and super intelligent. So what I'm saying though is that you do have mutations that get through, but most of the time they are caught by these spell checking mechanisms, and this is just one of them. There are many others. Let's talk about another way that mutations can get caught, or can become part of DNA, and that is from when DNA gets damaged. And so, if you have ever studied physics, where you've been talking about X-ray types of X-rays and X-ray based studies, or if you go to the dentist, for instance, and they take X-rays of your jaw, of your mouth, they make you wear that big lead vest, right? That big heavy lead vest that's hard to breathe on it's hard to breathe when you have this thing on your chest. But that is to prevent X-rays from going into as much of your body as possible. And you'll notice that the nurse or the doctor or the dentist always leaves the room when they push that button to take that X-ray shot. They always leave the room. Why do they do that? It's not because they don't like you, which I finally cleared up just the other day when I went to the dentist. Like, why do you always leave the room? I'm kidding. No, but they do that because they are if they're exposed to X-rays all the time, then they can sustain damage to their DNA. Of course, the nurse is not going to tell you, or the doctor's not going to tell you, mind if I leave the room to protect my DNA? They don't say that, but the thing is though that your DNA could be damaged by these X-rays. And so that big lead vest that you wear protects as much of your body as possible. And I guess your head is the only thing that's exposed, which doesn't really make much sense to me sometimes. Anyway, but still, so you have damage to DNA, and so the types of damage that you have here are where, for instance, you could have a double break in your DNA, where the DNA becomes weak on the rungs, or not the rungs, but the sides of the DNA, and that could happen by maybe an X-ray particle flying through, and somehow at the same point in the DNA or something lacerating both sides so that the DNA actually comes apart. That can actually happen. You can get what's called X-ray poisoning, or you can get radioactive poisoning. And that's because you have these particles, these charged particles, that just lacerate your DNA, and it can actually just come apart. You'll notice that people who do have like radioactivity poisoning, their skin looks like it's been burned, and that is because the skin is unable to be repaired. Your skin gets wear and tear, but the DNA that's supposed to contain the information about how to build new skin isn't available anymore. It's gone. So the skin just gets worn down, and then your skin begins to die because there's no regulation happening. And so that's why we see people with radioactivity poisoning. They tend to look really, really, really sick. Like they look like they've had a really bad, chemical burn or something. Well, it's because the DNA is no longer present to begin the repair processes. It's extremely important that the DNA remains alive. Anyway, so that's like a laceration here. You could also have maybe, for instance, this could be actually here we are. So these are X-rays from the double break. You have chemical bond between the neighboring nucleotides breaking up, and that's from ultraviolet light. If you're doing if you're doing experiments where you have an ultraviolet light in your workstation, you're typically supposed to wear a face shield, and you're supposed to wear a gown. And that prevents that ultraviolet light from actually getting inside and mixing up with and destroying your DNA. The DNA is very delicate. It's built tough, but it's very delicate. It doesn't do very well with some of these things. And if it's exposed to ultraviolet light, then it can break. And then sometimes it's when I say it breaks, it's rendered inoperable, but sometimes it's still being used by the cell, and so then you get cancers. Cancers or cells are still using DNA to grow, and the DNA has lost the instruction part that says, stop growing, we're done. The cell will just keep on growing, and you get masses that appear. That's where types of cancers happen. This is something that I worked on here, which is called, on the left-hand side, chemical modification of a nucleotide, reactive oxygen species, or ROS. That's one of my areas that I was working on. But these are kind of chemical, or they're like chemical burns, but there's no chemistry, I guess, in some senses. Or it's like you're burning your DNA without necessarily a flame. And so it's like it's environmental stress, like getting too much. Or not getting enough, I guess, enough rest, or not getting enough food, or not getting enough oxygen, or having too much salt delivered to your cells, or maybe not having enough insulin, or having too much insulin. Those things create what they call environmental stresses, and those environmental stresses actually do push your cells over the edge. They're trying to survive as well as they possibly can. The proteins are being pushed as far as they can go, and they just break. They cannot deal with this environmental stress. Microgravity was my area. If you go into space, for instance, because you don't have gravity around you your cells. Interestingly, your cells use gravity to navigate around. That means the thing that fixed the cells use gravity inside the cells to navigate their way around the cell. That means that when something happens to your cell, the nucleotide, the nucleus has torn, for instance, something comes along and fixes that. How does it find the nucleus? How does that thing find the nucleus? How does that thing find the DNA? How does whatever the thing is that fixes, or whatever that piece of biological machinery that has a job to do in the cell, how does it get there? Well, it uses gravity to find its way. That's like your GPS. It just knows that when I go up here, there's less pressure. When I go down here, there's more pressure. So that's kind of how it moves around. It's like your GPS for your machinery inside your cell. And when you're in space, your cell doesn't have that gravity, and so the pressure is like all over the place. Either sometimes there's high pressure up here, and sometimes there's low pressure down here, and it doesn't. These pieces to do these repairing jobs, they just get lost. And so when you go into space, when you come back, it's like you have aged much more quickly than you would have aged if you had just stayed on Earth. And so those people who are saying that they want to go into space and live up there, look at their faces when they come back. It's like their faces are older. Even if they've been up there for like maybe just a year, it looks like they've been up there for maybe 3 years. And also, though, I mean, just I'll tell you, when you see these these astronauts when they come back to Earth, when they come back, they're unable to stand. You see them sitting on deck chairs in front of their rocket ships when they come back to Earth. I mean, we all see that. Oh, it's a hero's welcome. They're all back, and NASA's gone out there to say hello to them or to welcome them back. And they pick these guys up, who are the astronauts, carry them out of the ship, or out of the spacecraft, I guess, and put them on these deck chairs, and they can rest there while they catch their breath, because it's hard to breathe. And then they are loaded into ambulances, and they go back and have their hero's welcome. Ambulances pick them up from space travel. Well, that's because their body has broken down when they're in space. The mechanisms that repair your muscles, the mechanism that repairs your heart, your lungs, your skin, your leg muscles, those mechanisms basically misfire when you're in space. And so when you come back to Earth, you can't move, you can't do anything, and you can hardly breathe, and you panic. I know this because actually I spoke to, when I was at the University of Nebraska, there was a Nebraska astronaut who came back. He was in space for not more than 7. No, I'm sorry. He was up in for not. I think it was like 13 days. So it was like a spacewalk he had, but it was like 13 days of time in space. That was it, just 13 days. And he said that when he came back to Earth after 13 days in space, it's an unlucky number, but when he came back though. He came back and he was sitting in his harness in his seat. They came and I think they crashed into the ocean or something. But they're just floating around, and somebody came out to pick them up. And there was this voice that came through saying, "Don't panic, just stay where you are. We're coming to get you." Like this really kind of calming voice. "We're coming to get you. It's okay. We will come inside and actually undo your seatbelt." And he's like thinking, "What do you mean undo my seatbelt? I can do my seatbelt." And he's like, "God, I just," and he couldn't do his seatbelt. He's like, "Well, that's okay. I'll just start wiggling my feet and get out of here, right?" And he couldn't move his legs either, and that was 13 days. And one of the things about him, though, is that he was also a track star. He used to run track all the time, and he said that it took him like maybe 6 months to a year to be able to like get to the point where he's able to do hurdles again. And he's like, "It's really embarrassing, but I used to pride myself on running hurdles and track, and I just couldn't clear the hurdle. I mean, I have my family watching me as I'm running track. I just couldn't clear these things for 6 months to a year." He had a physical therapy. So that was 13 days. And so here's this, let me just say something else here, and you guys kind of pick a thing. But when you have these people who are talking about, we're going to go colonize Mars. I'm going to be in space for 6 months. I'm going to be traveling to Mars in 6 months. It's going to be great. I'm going to be the first American in Mars. Great. Well, the thing is, though, that when you arrive in Mars, you're going to be so weak that you can hardly even pick up the phone to call anybody to tell them that you've arrived, because your muscles will break down. Because when you're exposed to microgravity, you have all kinds of damage to DNA that happens, and you just can't move your arms. You can't move you can't move your muscles, because the muscles cannot be repaired as effectively as they would be on Earth. So anyway, that's a very important piece of biology to understand if you're going to be traveling in space. And so I say, stay on Earth. Earth power, man. I'm a total Earthling. Come on. Anyway, so these are just like pieces of damage you can get for DNA. Here we have chemical linkage between two strands, where you have like another reactive oxygen species. This basically means that you're subjecting the DNA or the protein or whatever it is, that the whole thing, I guess, but in this case the DNA. You're exposing the DNA to types of environmental stresses. Maybe it's heat, maybe it's salt, maybe it's something else. But the DNA doesn't like that, so it does these bad things. It's like if I took this computer and I put it in the oven, and I turned the oven up to 400, and I let's let it go there with a pizza. The pizza might cook because the pizza's okay with that, but the computer doesn't like heat. It's going to melt. And the same thing could happen to your DNA. It's going to melt when it's exposed to some of these types of environmental heats and stresses. So you have mutations from the environment. Anyone ever heard of like sunspots and those kinds of things? These are like storms that happen. There's like lots of radioactive, storms and radioactive energy that's flying around all over the place. And you have like, solar winds, you have radiation, you have all kinds of things in space which are really hostile. By the way, has anyone noticed that there is actually an orange on these slides here? As scientists, you're supposed to notice everything. You'd be surprised. Actually, this slide, I've changed the text, but that picture actually came from a conference that I gave, where I was talking about reactive oxygen species, which is right down here. I was actually talking about that as part of my own research, and nobody noticed that there was an orange in there. And I thought, why did anyone not notice this? I mean, you guys are all scientists, right? Right? Okay, but anyway, but you have these environmentally caused mutations, which is like ultraviolet light. That's why they're saying that when you go to the beach, you should like put on that sunscreen, save your skin. And the reason for that is because if you don't have any protection on your skin, then you have this ultraviolet light that comes from the sun that somehow got through the ozone layer. It hits your skin, it goes inside your skin, and it goes inside to the DNA inside the light will make it, or the ultraviolet light will make its way to the DNA, and it will cause these kind of double connections to happen between, or these double bonds to happen between the T's of your DNA, like in this case. These cause these are what they call thymine dimers. A dimer is like where you have a kind of a loop where the DNA kind of loops on itself, and so. It doesn't sound like that's very bad. I mean, the DNA hasn't actually been removed or changed or whatever it is. It should be an easy fix. But actually, though, it's sometimes difficult to undo those TT bonds in this kind of scenario, and so you get you can get some problems with your DNA. It actually causes damage. So when the DNA is being fixed, there's a guess that has to happen here. The polymerase here has to guess how the DNA looked before the damage from the ultraviolet light. And if that guess is wrong, that means the DNA is going back wrong, which means your proteins are now going to be getting the wrong amino acids. And by the way, let me ask you this. Does anybody here know why your skin peels when you go to, and the same thing with radiation poisoning, but why does your skin peel when you go to the sun? When you have like a really bad sunburn, your skin just comes off in sheets, it seems? Anyone have any ideas why that would be? No? Should I tell you? Yes, sir.
I just thought it was like top layer of your skin like dies.
Yeah. But why would that's true? So that the top layer of your skin does die. But why is that why is that an advantage? Why would you want your? It takes energy to build that skin back. Why would you want to? Why would your body just make the call and say, kill the skin on my arm? It's, what do you think?
There's like dead skin on the top, and it protects like the lower layers that are still living from being burned.
Partially, but it's more fundamental than that. What were you gonna say?
The body like doesn't want to risk them mutating.
Yes, the dead skin does protect. It does protect. That's part of the strategy, but it's more or less. I think it's that the body doesn't take any chances with broken DNA as far as it can. It says this DNA is damaged. I know that there's been some kind of ultraviolet light that has been hitting this DNA. It could be damaged. Maybe it's safe. I don't know. I'm not going to risk it. Just scrap it all and rebuild. And so that's why it comes off, because you have what's called apoptosis or self-induced cell death, where basically your cells are killed off by your own body because they are thought to have bad DNA. And if the DNA is bad, then that could make a whole bunch of other problems. And so rather than risking it, it just says it's easier to rebuild the skin. When you have a sunburn, though, it's like your skin will just come off. But if you burn your skin with water, if you have a steam burn, you get blisters, and that's kind of the same idea, that the skin is again being killed off because the DNA was subjected to this environmental stress of heat. Maybe it's okay, maybe it's not. I don't know. I'm not going to risk it. I'm going to just rebuild all my skin. And so that tends to be what your body does. That the cells will have to die for. Just so that you have the security of saying, if there was any damage that had happened to this DNA, then I think that we got it. It's not there anymore. I think that we've taken care of that problem if there was a problem. But the dead skin does serve in some cases to protect the old stuff. But it doesn't last forever, though, because eventually it gets worn off, and so you have that. Anyway, but so still, you do have this, you do get rid of this stuff if it seems like there's an error. Now, let me just talk about something else which is actually quite interesting. This is what they call a silent mutation. And by that, so far I've been saying that if you have a mutation in your DNA, that DNA is then used to make is used to make a piece of protein, and if there's a mutation in the DNA, then the protein is then going to be misformed. But one thing we talked about last week, I don't think I have it here, but one thing we talked about last class I think it was that there's often more than one way to make an amino acid. And what I mean by that is, like, for instance, you have more than one type of codon that can create the same amino acid. And there's this high level of redundancy. You know, I'm thinking of like leucine, this like has, I think, 8 different codons or 8 different combinations of RNA that can be used to create leucine. I think that's 8, but anyway, it's more than one. And so in this case, you have a codon TAT, which creates this, was it tyrine here? Well, that's called T. But you have this TAT, which creates this protein amino acid here, TYR. And then if you somehow changed the C, or the T at the end of the codon, the T becomes a C, then you still get the same protein. You still get the same thing. The same protein still is still created. It comes from a different codon, but it's still the same thing. And so for a long time, a lot of researchers thought that, well, it's not really a mutation. It's just, you still get the same protein, so we're all good, right? But actually, though, they call this a silent mutation, though, because you don't know whether, for instance, how that DNA is actually being read. And what I mean by that is that you sometimes have what they call nested genes, where one gene is read on, when you start at base 1, then you read base 2, base 3, and that's one of your codons. And then you continue the cycle as you keep on going down the gene. But you may have another base here, or another gene that's nested, where, for instance, this first base, you don't start at that, but you start at the second base, and then you start reading in 3s, starting from base number 2. And so then you have this nested ability here. So you're reading the same piece of gene, but you're just frame shifting yourself in some ways so that you're getting a slightly different protein that comes out of it, which is actually very exciting. When you see that kind of thing happening, it's very, very exciting. But the thing is, though, that if you have a silent mutation in that kind of situation, then your nested gene is probably going to get a different type of amino acid. So it's not exactly like they say silent. By that they meant it's like, you don't know what kind of mutation it is. You don't know whether it's dangerous or not. And so you don't always know whether that's a dangerous thing or not dangerous thing here. But the thing is that the mutations will occur in a gene, but you don't always see that there's not a clear message of that mutation ever having happened. They say that this happens, they say that protein coding regions make up only approximately 1% of the human genome, which means that you don't have, there's a lot of places where this thing couldn't be dangerous, but then there's this 1% of the genome, which actually does code for protein, where if you have this mutation in that 1% area, then it could be very dangerous. So yeah, one thing I should say, though, is that before we get into the sickle cell anemia, is that these mutations are actually random. It's not like, for instance, the mutations just kind of make their way to only the coding regions or only the other regions. They're random. They happen anywhere. And if they tend to happen in a place where the code is making protein, then that's bad. But if they happen to in an area where it's not making code, or it's not making protein, then it's not so bad. So we say. But of course, more research is always necessary to find out whether these so-called non-coding regions are really non-coding. And they say that they're there for a reason. They're there as spacers, for instance. They help the DNA to be wound up into chromosomes. And so maybe having a mutation there would somehow interfere with that. Anyway, sickle cell anemia is actually a very dangerous and a very unfortunate disorder, which happens when the red blood cells are basically given the wrong protein, like one protein here, which is just changed, and the cell, the red blood cell, is no longer like a round pillow thing that kind of floats easily through the veins here, just like a whitewater raft that like nicely, easily bounces off things and kind of floats through. But rather, the new cell is like the sickle, and it gets stuck on things, and it pokes things, and it gouges things, and it causes like blocks and things. You can see how, for instance, in this normal capillary, which is a small vein, which is basically wide enough only for one cell, how if you're a round object, you're easily able to kind of like skirt through these areas. Just like if you're whitewater rafting. If you're on a round raft, it's easier to get through. But if you're on a raft that has some funny shaped, some funny shaped thing here that has like all these pokey things and spikes that come up, those will get caught in things, and that causes blockages and strokes. And so it's very, very dangerous. Well, you're probably thinking, how do you get this? Well, in this case, it comes from a mutation, a simple mutation, where, for instance, you have the mutation has it's like a point mutation in this case, where you have the wrong protein being created. So you have this G, which is glue, the glue word, and then you have valine, I think it's valine, but val, G and V, are somehow they're not you have the wrong protein being created. So in this case, you have GAG on one side, and that A in the middle becomes a T. Well, the thing is though that T and A still attract each other, they still bond with each other, but it's not supposed to be that the sense strand has the T and the antisense has the A. It's supposed to be the other way around. And so while that's a very subtle mutation, it has very grave consequences. I think that with that, we're probably running out of time here. Yeah, we're almost, we have like 1 minute left here. I think I might stop there. Actually, I will stop there. When we come back on Monday, we are going to be talking about, we're going to be using something called Biopython. And so we're going to be doing some coding here. So again, bring your computers, make sure that your Docker is working. And by the way, I've changed your Docker file. Every time we have when we do coding here, I will have to change, if I have to change things, I will. But I've changed your Docker file so that there's a new line in there that actually installs something called Biopython. And so please be sure that you know how to use Docker when we come to class, because we have to rebuild that same container to run the new code. All right, looking forward to it. Very exciting. Everybody have a great week. Stay well.
Thank you.
Thank you.
Thank
Take
you,
care.
Professor.
Bye-bye, guys. Be well.